The following method offers reliable and sensitive detection of specific chromatin modifications on selected plant genes. First, cross-link modified histones and DNA with formaldehyde. Then, extract and sonicate the chromatin to facilitate immunoprecipitation with modification-specific antibodies. Finally, analyze the chip reaction by the decross-linking of histone DNA complexes and gene-specific real-time quantitative PCR. In plants, this approach can provide molecular insights, like the specific histone modifications associated with C4 photosynthesis in maize, and systemic immunity in Arabidopsis.
The main advantage of chromatin immunoprecipitation over existing methods like mass spectrometry is that this technique allows the detection of histone modifications on selected sequences in the plant genome.
Generally, individuals new to this method will struggle because the protocol entails a couple of steps whose correct performance is key to success of the procedure.
In this video, we use chip analysis to provide insight into the regulation of gene expression in the model plant Arabidopsis thaliana. Demonstrating the procedure will be Michal, Jaskiewicz, a PhD student.
For each sample, begin with a harvest of 2 grams of Arabidopsis leaves in a 50 milliliter plastic tube. Fill to the 40 milliliter mark with cross-linking buffer and position a tailored filter sponge right above the buffer surface to ensure the leaves stay immersed. Now place the samples in a desiccator with vacuum infiltrate for 10 minutes. To stop the cross-linking reaction, add 2.5 milliliters of 2 molar glycine and mix by inversion. Then infiltrate again for 5 minutes. Continue to